Give me a second. Okay. By now, probably we are live. Sorry about the delay. Gmail wanted to verify that it's me. So hello, everybody, and welcome to another session of our Sussex Vision seminar series, as always within the Worldwide Neuro Initiative. I'm George Kafedis, a master's graduate from Thomas Euler's group, and currently a PhD student with Tom Baden. And as your host for today, I would like to once again begin by thanking Tim Vogels and Panos Bozelos for putting forward this ever-expanding initiative towards a greener and much more accessible seminar world. Having said that, allow me, of course, to get back to the reason we all gathered here for today and introduce our guest, the director of Max-Planck-Institute for Biological Intelligence, Professor Alexander Borst. Axel studied biology at the University of Würzburg. During his PhD with Martin Heisenberg, he worked on the olfactory system of Drosophila and demonstrated the indispensable role of the mushroom body in olfactory learning. If people hear us fine, just let me know, please, because I don't get any feedback from the second screen. But in the meantime, I will continue. He then moved to Tübingen and the Max-Planck-Institute for Biological Cybernetics for postdoctoral years with Carl Goetz. And in 1993, if I'm not mistaken, he became a junior group leader at the Friedrich Miescher laboratory of the Max-Planck Society. Following a couple of years as a professor in UC Berkeley, in 2001, Axel became the director at the Max-Planck-Institute of Neurobiology, as it was named then, nowadays Max-Planck-Institute for Biological Intelligence, located in Martinsried in Munich. After his PhD work on olfactory systems, and while still exploiting the advantages that the Drosophila offers as a model organism, Axel shifted his focus on a different neural computation, visual motion. And throughout the years, his work has been instrumental in deciphering how this computation is performed, both at the single cell and at the circuits level. For experiments in his lab, they employ a wide array of genetic tools, IFIs, two-photon imaging, and behavioral assays, while modeling plays an integral part in bridging theory with findings. So in his talk today, we have the pleasure of hearing about their latest work on the topic that has fascinated him since his postdoctoral years, namely how fly neurons compute the direction of visual motion. So without any further ado from my side, please all welcome Professor Borst. Axel, the stage is officially all yours.
Okay, so I hope you can all see the screen. Thank you very much for the invitation and for the kind introduction, George. It's a pleasure to, present here at this Zoom meeting. And as George already mentioned, my interest is in general on computational neuroscience. That is, how neurons perform an interesting computation on the signals that they receive from a sensory organ or other neurons, and then process these signals to come up with an interesting and important output quantity. And I'm interested in that phenomenon, not just at the circuit level, which neurons are doing the job, how they are connected, and which sort of neurons constitute the circuit, but eventually also at the biophysical level, which transmitters are used, which transmitter receptors are involved, which ion channels, and all of that. And so I really want to understand that computation, so to say, in depth. And as you will see, the question how fly neurons compute the direction of visual motion is really a practical example for neural computation, and it's been extremely successful and satisfying in the last years to get at this question at that level that I always wanted to get at. So let me start by introducing to you the fly nervous system. So here you see the fly nervous system in blue, and if we zoom in into the brain of the fly, you see that large part of the brain is taken up by the optic lobes that are shown here in red. The optic lobes, the analog of the vertebrate retina, is built from several layers of neuropil called the lamina, the medulla, the lobula, and the lobular plate. And as you can see, the columnar structure of the eye is repeated by each of these neuropils. And so they are built from these repetitive columns, these repetitive units that are arranged in a retinotopic way, such that neurons in neighboring columns process the signals that come from neighboring places on the retina and thus from neighboring image points. This columnar structure is then given up at the level of the lobular plate, where large tangential cells collect the signals from the columnar neurons with their large dendrites. And here is right away the phenomenon that we want to study. If you move a bar in front of the fly's eye to the right and to the left again, and you record the signals of a single photoreceptor, then you see the same signal each time. And clearly, the signal of a single photoreceptor is non-directional. You cannot tell the direction of motion from looking at the photoreceptor signal. Now, if you take this electrode and just move it a few synapses downstream and stick it into one of the tangential cells, the situation is very different. Here, the signal is clearly directional. These cells depolarize to rightward motion, that is called the preferred direction, and they hyperpolarize to leftward motion, that's called the null direction. So within a few steps, you go from a non-directional to a directional signal. And so that's a phenomenon where they compute the direction of motion in between. And how is that done? Well, this is exactly what has been proposed by Bernhard Hassenstein and Werner Reichert in the more than 60 years ago. They did behavioral experiments on a beetle walking on a spherical Y maze and presented this beetle with various patterns moving to the left or to the right, doing apparent motion and all of that. And they came up with a model that extracts the direction of motion from the input signals to this beetle eye. And this here shows the model as it was originally published in '56. And here you see the model, the essence of the model. And this model is built from two mirror symmetrical subunits that share the same two input lines. And within each subunit, the signal from one image point is delayed by a low-pass filter with the time constant tau, and subsequently multiplied with the instantaneous signal from the neighboring image point. And that is done twice in these subunits. And then finally, those outputs of the multipliers become subtracted, and that leads to a direction-selective signal, just as we've seen in the signal of the tangential cells. And this motion detector model not just reproduces qualitatively this phenomenon, it also does so in a quantitative way. And I spent much time as a postdoc together with my colleague Martin Egelhaft to challenge this model by doing all kinds of computer simulations and stimulating it with various patterns, and then comparing the computer simulation output with the signals in the photos of the tangential cells. And each time it convinced us that this is sort of somehow implemented. This motion detector model describes the processing of the signals within the fly optic lobe between the photoreceptors and the tangential cells. So, of course, the question was then which cells are doing this job? And so let's take a look at what's in between. And we take a horizontal cross section through the fly optic lobe in order to see the different cell types that there are. First thing you notice are the photoreceptors are 1 to 6 that send their signals into the lamina, the first neuropil. And there, the signal is taken up by lamina monopolar cells, L1 to L5, which then send the signal to various strata in the medulla. Within the medulla, you find intrinsic medulla neurons called MI, that link different layers of the medulla, and transmedulla neurons that take the signal from the medulla to the lobula. And of particular interest are two cell types, namely called T4 and T5. T4 cells have their dendrite here in the medulla layer 10 and ramify within one of the 4 layers of the lobular plate, and T5 cells have the dendrite in the lobula and then have the axon terminals in one of these layers. And these will be sort of very important cells for the remainder of the talk. These are just, example cell types. The full account of all the different cell types that exist in the fly optic globe were given by Golgi stainings from Karl Fischbach and his student Dietrich, and were published in '89. And there are about 100 different cell types in the fly optic globe. And each of them here is shown once, but you should keep in mind that they exist once per column. And unfortunately for us as the community interested in fly motion vision, all these cells were way too small to record from them at that time. And so the visual response properties of none of them was known. That was quite unsatisfying, because on the other hand, we had this perfect model that described the transformation of the input signals to the output signals so well, but the relation between the two was unknown. It was like a black box. And thanks to Drosophila and the genetic tool set that it offered, we could sort of open this black box in the last few years. And using the genetics of Drosophila, we were able to make a number of important discoveries. First discovery was that motion is not computed once, but in parallel channels, one in an off channel and one in an off channel. Second major discovery was that T4 and T5 cells represent the primary motion sensitive neurons, T4 in the on channel, T5 in the off channel. And finally, a new cell type, the LPI neurons, were discovered that implement the subtraction stage of this motion detector. So let's talk a little bit about the T4 and T5 cells. Like every other neuron on Earth, they were first described by Ramon y Cajal. And Ramon y Cajal for 2 years worked also on the insect nervous system and published this book in 1915 together with Sanchez. And there you find nice drawings of T4 cells with the dendrites here, axon terminal there, and T5 cells with dendrites here and axon terminals there. But they were sort of a cell type or morphology that he wasn't used to see. So he called them curiosos elementos generadores de tallos bipennachados, which means strange elements that generate B-tufted sticks, so to say, the dendrite and the axon terminals like on a stick. So he couldn't sort of figure out the information flow, so to say, like he did with pyramidal cells and all of that. So meanwhile, we know that there exist exactly 4 subtypes of each T4 and T5 cells per column. And each subtype ramifies within one of the 4 layers of the lobular plate. If we do a single cell flip out genetically, then here you see the dendrite of the T4 cell, here you see the soma of the T4 cell, and this is the axon terminal. If we take a look from the top, we see that the dendrite of a single T4 cell covers actually 7 to 8 columns in the medulla. And then if we stain the somata and count them in each side of the brain, we come up with the number of about 6,000, which is exactly 750 times 8. So these are the cells. What are their response properties? Well, importantly, we got about 10 years ago, or exactly 10 years ago, we got a driver line for the T4 and T5 cells from Gerald Rubin and Alyosha Nern at Genelia, and that allowed us to do 2-photon calcium imaging from these cells and stimulate them visually. So this is the driver line, and this is a GFP staining, and you see the dendrites of the T4 cells, you see the dendrites of all the T5 cells. This is the inner chiasm and you see the 4 different layers of the axon terminals of these cells. And this is how they look in the confocal. And under the 2-photon microscope, they look like this. And you see already structurally 1, 2, 3, 4 different layers. And now Matt and Jürgen in the lab presented visual stimuli to these flies and then measured the relative change of fluorescence. And when they stimulated the flies with grading moving from the front to the back, they saw a very strong activity in layer 1. If they moved the grading from the back to the front, they saw a very strong activity in layer 2. When they stimulated the flies with upward motion, they saw activity in layer 3. And when they stimulated it with downward motion, they saw activity in layer 4. And now, if you assign a particular color to each pixel according to which direction it responded the strongest, then you see these 4 beautiful layers that correspond exactly to those 4 anatomical subtypes that I told you before. So subtype A, projecting to layer 1, respond the strongest to rightward motion, this one to leftward motion, to upward motion, and to downward motion. So this is very nice. But the next question, of course, was, was a driver line for T4 and T5 cells. What is the functional difference between T4 and T5 cells? And for that, we got, again, from Gerald Rubin and colleagues, specific driver lines for T4 cells and for T5 cells. And we then applied 8 different stimuli, namely dark and bright edges moving to the right, to the left, upward and downward, in order to see their preference for the contrast polarity. And what we found was that the T4 cells respond specifically to on edges and very little to off edges. And so they are the motion sensing neurons in the on pathway. And the opposite is true for T5 cells. T5 cells very strongly respond to moving off edges, dark edges, and very little, if at all, to moving on edges. So that was sort of a very important step for us in the lab to discover that T4 cells are the primary motion sensing neurons in the on pathway and T5 are the primary motion sensing neurons in the off pathway. Now, the question was then whether these neurons are the motion detectors or whether there exist other motion sensing neurons in parallel to T4 and T5 cells. And one good way to testing that is through behavior. And so what Armin Bahl, Georg Ammer, and Tabea Schilling did in the lab was to test the optomotor response of these flies with T4 and T5 cells blocked. And so they put a fly on an air suspended ball and surrounded them by gratings moving in one or the other direction. And here you see the optomotor response to clockwise motion and counterclockwise motion of control flies. They always follow the grating motion. And that is true for all contrasts above maybe 5 or 10%. So this is the contrast dependency of control flies. Now, when we block the T4 and T5 cells and then test the flies, we get a straight 0. So this is the optomotor response of T4, T5 block flies. It's 0, and it's 0 throughout the contrast tested. So this is very amazing, and it's a very strong effect that we see. But maybe we thought that's just the sort of large field response, that's the optomotor response. Maybe there are other responses that are driven by other neurons, not T4 and T5 cells. And so we tested the response to a single bar moving through a window on the right side of the fly. And here, Armin, Georg, and Tabea applied 3 different stimuli. The first one was to move the bar from the front to the back through this window, then from the back to the front through this window. And then, that's very interesting, they just applied a luminance modulation that mimics the luminance modulation occurring during a moving bar. But of course, this luminance modulation, you know, the window just becomes dark and bright again, has no directional information. So first, the responses to of control flies to these different stimuli. You see a very strong response to front to back motion, very little back to front motion, an intermediate response to the luminance modulation. Now, when you now test the T4, T5 block flies, you get the same response each time. And you know, this is the red trace. And I find that extremely interesting because it tells you that to these T4, T5 block flies, the stimulus is always the same. They just perceive the luminance modulation, nothing more. There is no vector, no direction to the stimulus anymore. And so we also tested other directional responses of the flies, like the landing response and avoidance response and all of that. And every time in T4, T5 block flies, the response was 0. So from all of these experiments, we conclude that they are completely motion blind. So yes, indeed, T4, T5 cells are the primary motion sensing neurons in the fly brain. So that now brings me to the question, how do they become direction selective? How do they do it? What is the biophysics behind that? So there were basically two mechanisms proposed in the literature to account for direction selectivity. And one is called preferred direction enhancement. The other is called null direction suppression. For preferred direction enhancement, you take the signal on one side of the motion detector, and you pass it through the low pass filter, and then you enhance the signal from the central input here. And a multiplication has been proposed for that. This is the Hassenstein-Reichardt idea that I just proposed to you. But there was also another idea from Barlow and Levick doing experiments on the rabbit retina in the 60s of the last century, and they proposed null direction suppression. So in this case, you take the signal on the other side of the central input, again delayed, but now you suppress the signal here. And the operation proposed for that, also by Tommy Poggio in later sort of analytical studies, was to propose a division for that. Now, in order to discriminate between preferred direction enhancement and null direction suppression, Jürgen Haag used a telescope to place exactly the light flashes onto the center, to the left and to the right within the receptive field of an individual T4 cell. And later he repeated this experiment with T5 cells. And what he found was the following. If he preceded the central flash with the flash on the left side, then he saw an enhancement of the response to the central flash. So that speaks for preferred direction enhancement. But if he preceded the flash with a flash on the right side, he saw a suppression of the response to the central flash. So that clearly spoke for null direction suppression. So the conclusion was that we are not dealing with one or the other, but rather a combination of both mechanisms. And this gave rise to this sort of three-arm motion detector, which was also confirmed by studies from Tom Clendenin's lab in the same year. So this seems to account for direction selectivity in T4 and T5 cells. So next question, of course, is which cells are the input neurons to the T4 and to the T5 cells? And thanks, no, first I want to say what is the advantage of such a dual mechanism of direction selectivity. And here you see the directional tuning of a full three-arm detector where you have A times B divided by C. And when you now sort of in simulations move the grating in all possible directions and then you plot the response strength in such a polar plot, you see a very nice tight directional tuning of such a motion detector. And each sub-process by itself still gives you directional tuning. Like this one is also tuned to rightward motion, but is broader than the full detector. And the same is true for just null direction suppression. There you also have a nice directional tuning, but you have responses to null direction motion, which you don't have here in the full detector model. So the combination, I think, sort of the two mechanisms working together leads to this nice tight directional tuning of the motion detector. Next question is, of course, which cells are doing it? And here we profited again from our colleagues at Genelia Farm, who provided us with the connectome of the fly optic lobe from EM analysis. And we now know that T4 cells receive input from these 6 cells called MI9, etc., etc. You don't need to remember all the names. And we also know the inputs to the T5 cells. The important thing is that they don't sort of collect the input from the same cells in each column, but depending on their preferred direction, they do so in a spatially selective way. So the T4A cell, tuned to rightward motion, collects MI9 in the left column, MI1 and TM3 in the central column, and MI4, CT1, and C3 in the right column. Now T4B, tuned to leftward motion, does it in a mirror symmetrical way. They get the same central inputs, but T4B collects the signal from MI9 on the right side and MI4 to C3 on the left side. So what is very nice that came out of this EM analysis is not just sort of identifying the input neurons, but also seeing that this 3-arm structure that we postulated from functional experiments is reflected in the anatomy, how they collect the input from their input neurons in a spatially selective way. As I said, the same thing we find in the T5 cells. Here, there are other neurons, but again, it's spatially selective depending on the preferred direction of the T5 neuron, the subtype. So now we did functional analysis on these neurons. And for the sake of brevity, I'm just talking about 3 of the input neurons, sort of representatives. It's MI9, MI1, and MI4, right? And that's true, you know, in this spatial order for a T4 cell that is tuned to rightward motion. Now, Alexander Ahrens, Michael Drews, Florian Richter, and Georg Ammer in the lab did calcium imaging on these input neurons. They stimulated the cells with white noise and then did reverse correlation to determine the spatial temporal receptive field. And this is what they got. Now, as expected, MI1 and MI4 have on-receptive fields, in this case surrounded by an antagonistic surround, an off surround. And the width of the receptive field is about 6 degrees. So it's about, you know, 1 column, what you expect from one single facet in the fly's eye. But amazingly, MI9 turned out to be an off cell surrounded by an on field here. And that will become important later. Now, looking at the temporal properties, we found that MI9 and MI4 have low pass characteristics. So what is shown here is the step response, whereas MI1 has a band pass characteristic. And that nicely fits to the placement of these cells on the dendrite of the T4 cell. As I said, you know, the flanking neurons should have low pass characteristics, whereas the central one should be fast. And this fits exactly the temporal properties of these different neurons. So we can recreate the directional tuning of T4 cells formally by just taking the signal of MI9, sign inverted to make it an on, multiplied with the signal of MI1 and divided by MI4, and here we go. So that's very satisfying. But as I told you, we're also interested in the biophysics. You know, this is just an algorithmic description, A times B divided by C. Now, what I want to get at is replace that by an electrical equivalence circuit, where the signals actually change conductances of the postsynaptic dendrite and then lead to that. And as a first step towards a biophysical model, we and others identified the transmitters of the different input neurons. And what came out of that analysis is that MI9 is glutamatergic, MI1 is cholinergic, and MI4 is GABAergic. Now, transmitters is one thing. The other thing is the transmitter receptor on the dendrite of the T4 cell. And thanks to our single cell RNA sequencing technologies, again, we and others looked at the transmitter receptors in the T4 cells, and we found the following subunits expressed. Various nicotinic receptor subunits, GABA A receptor subunits, mainly RDL, and then something very interesting, and that is the gluCl alpha receptor. And that is very interesting because the gluCl alpha receptor is a glutamate-gated chloride conductance, which makes the input neuron inhibitory, the glutamatergic input neuron. And so, the idea was immediately that, since MI9 is an off cell, maybe through inhibition, it does the sign inversion required for the on channel here. But before, going into the details here, we were also interested whether the receptor localization on the dendrite matches the placement of the different input neurons on the dendrite that was determined in connectomics. So here you see EM reconstruction from Genelia farm of the different subtypes. And you see that the different subtypes of T4 cells, A, B, C, D, have their dendrites oriented against their preferred direction. So always with the tips where the preferred direction stimulus is coming from, T4A to the left, T4B to the right, and T4C downward for upward motion, and this for downward motion. Now, Sandra Fendel and René Vieira in the lab now tagged the different receptors by a specific technique called flip tag, and then endogenously allowed to visualize the different receptors in single cell flip outs. And what they found was indeed that the gluCl alpha is located at the tips of the dendrite, where it receives MI9 input. The nicotinic acetylcholine receptor is distributed in the center of the dendrite, where MI1 and TM3 provide the input. And the GABA A is concentrated on the base, where those three other neurons provide the input. And they quantitatively evaluated their data, and this is the result. You see gluCl alpha at the tips, the nicotinic receptor in the center, and the GABA receptor located at the base. Now, with this, I thought I can come up with a model, a biophysical model of direction selectivity in T4 cells. And having now MI9 control a glutamate-gated chloride channel, and MI1 an excitatory cholinergic synapse, and MI4 a GABAergic. And this is how it could work. If you have the visual scene in the dark, then MI9 will be active all the time because it's an off cell, and MI4 will be silent because it's an on cell. Now, if you flash a central flash to activate MI1, the input resistance will drop a little bit, and you will see a depolarization of the postsynaptic T4 cell. Now, let's consider movement of an on edge. In this case, the on edge will suppress the activity of MI9, and that will lead to a closure of the chloride conductance. This closure of chloride conductance will lead to an increase of the input resistance of the T4 cell, and hence the depolarization provided by the MI1 will be much bigger. So this can lead to an amplification of the response due to the decrease of MI9 input and an increase of the input resistance. Now, for motion in the opposite direction, you have the sequence of MI9 and MI4 will be such that they overlap. That means inhibition will be always active. That will lead to a decrease of the input resistance and hence to a smaller depolarization in response to the MI1 signal. So in this case, you will have a null direction suppression, right? So this is how it could work. Now, in order to test how it does work, we had to overcome one major obstacle. And this major obstacle was that so far, all the recordings from T4 and T5 cells were done, at least in our lab, with calcium imaging. But calcium imaging does not allow you to measure input resistance or reversal potential or anything like that. So following in the footsteps of Eyal Grundmann and Michael Reiser at Genelia Farm, they were the first that managed to record wholesale patch recording from T4 and T5 cells. And then a very brave postdoc came to my lab, Lukas Groschner, and he has golden hands and managed, within a few months to record from T4 and T5 cells like on a daily basis. And he taught a grad student, Jonathan Malis, and together they were sort of the Incredibles, and they recorded from T4 and T5 cells, or T4 cells mainly, and also from all the various input neurons of the T4 cells under identical stimulus conditions. So they created an incredible data set to work with. And so this is how it does work. Now for edge motion in the preferred direction, you see MI9 signal being suppressed, followed by MI1 signal, and that is then followed by the MI4 signal. But importantly, those two inhibitory neurons, MI9 and MI4, leave open this window of opportunity for the MI1 signal to depolarize the T4 cell, just like in the simulation you've seen before. So consequently, T4 depolarizes nicely and strongly by about 20 millivolts. And for null direction motion, the two inhibitory signals of MI9 and MI4 overlap, and so they close this window of opportunity, and hence the depolarization to null direction motion is very small. So this is very nice and provides sort of an explanation of preferred direction enhancement. But we were sort of critical and wanted to ask a few more questions. The first question was, is glutamate really inhibitory in these cells? And for that, the following experiment shows that yes, it is so. When we puff glutamate on the dendrite of a T4 cell, then you see a nice depolarization. If we block the CL alpha by RNAi expression, the cell is depolarized and no longer responds to glutamate. So glutamate is inhibitory. The next thing is whether can glutamate really control the input resistance of the neurons. So Lucas did the following experiments. He puffed again glutamate on the dendrite, but injected current at the same time. So without glutamate, you see a very nice, strong membrane depolarization and hyperpolarization in response to current injection. Now, if we puff the glutamate directly before the current injection, you see that the depolarization and hyperpolarization has a much smaller amplitude because of the diminished input resistance. So glutamate can control the input resistance of these neurons. Next question is, does the input resistance also change during motion? And so Lucas also measured the input resistance during these visual stimulation. And here you see a preferred direction response, the depolarization you've just seen, and the dashed line is the input resistance. And it starts off with the value of about 5 megohms and then rises to 8 megohms just before the depolarization. So the input resistance is increasing dramatically before the depolarization starts. And the last question is whether it has an effect on the directional tuning of the cells. And so this is the directional tuning of a T4 cell in control flies. And if we again remove glucial alpha receptor by RNAi expression, then you see that the directional tuning becomes less expressed, right? So it's still there, but it's less than in the control flies. So now that I think sort of clarifies the point of preferred direction enhancement in T4 cells and shows the biophysical mechanism of this multiplication. Now, there is, of course, the other process at work, and that is the null direction suppression. And how is that done? Now, here, important data were gathered by Eyal Grundmann, Romani, and Reiser at Geneva Farm. And what they did was to present flashes in different parts of the receptive field of T4 cells. And what they saw was that in the center, you have a nice and strong depolarization, whereas if you have it on the null side of the receptive field, you get a hyperpolarization. So this already indicates that null direction suppression is active, but to see the effect of null direction suppression for the directional tuning, Amalia Braun and Matthias Meyer in the lab removed the GABA receptor subunit RDL and compared the directional tuning in these flies with the one in control flies. And they worked on T4 subtype C cells that are tuned to upward motion. And the black line is the directional tuning of control flies, nice and tight, as you've seen before. And now when you express RNAi, remove null direction suppression by removing the GABA receptor, you see this broadening. So that's the impact of null direction suppression on the directional tuning. And together with preferred direction enhancement acting in concert, they give rise, both processes give rise to the nice tuning that you see here. So that now allows me to, or allowed me to come up with a T4 cell model, a biophysical model that uses the input signals that were measured by Lucas and Jonathan and were confirmed by behavioral experiments then with Birte. After adjusting the threshold and the slope of the transfer function of the various signals, it allowed me to sort of adjust the parameters and then get model responses that fitted the experimental responses in a quantitative detail. And here you see the data of T4 cells to preferred direction motion and to null direction motion for an on-edge moving at 15 degrees per second. And this is the model responses. And I think, it nicely fits. And the same thing is true for 30 degrees. Again, here are the data and here are the model responses. And then applying this model with these parameters to the whole data set that they created for gradings moving at 4 different velocities or 5 different velocities and on edges for 5 different velocities. The preferred direction responses are always in blue, the null direction in red, and in the fat colors is the data and the faint colors is the model. You see a very nice agreement between the model and the data. So I think we have a fairly good understanding of how direction selectivity is created in these T4 cells at the biophysical level. And for me, to get even a better intuition of how that works, I used a sound mixer and attached it to the computer. I'm also a hobby musician and have a little recording studio at home. So I'm used to mixers with sliders and, knobs and stuff like that. And so I programmed the simulation such that I could control the various parameters with the sliders, like these sliders for the gain of the 5 input neurons, for the threshold of the 5 input neurons, or here for the reversal potentials of the different ionic currents. And so I could sort of play around with that, and I can sort of block input signals by just mute them or solo them, and for the, this way build up a very good intuition for which cell signal has what sort of functional impact on the final response properties. So that's all very nice. So as I said, I think we have pretty much done with the biophysical mechanism underlying direction selectivity in T4 cells. So question is, of course, how about T5 cells, the on-pathway neurons? Well, here again, as I said already, we know the input neurons, and intriguingly, it's a bit different from what we see in the on-pathway. So we have one cell, the TM9 cell here on the left. We have 3 cells in the center, TM1, 2, and 4. And we have on the null side, 1 cell that is CT1, and that is the only GABAergic one. These are all cholinergic neurons. Now, CT1 then, being GABAergic on the null side of the dendrite, should be the one that provides null direction suppression. Now, that's sort of very interesting because CT1 is a huge amacrine cell. It exists only twice per fly brain, one on each side. And here you see the reconstruction done by Amy Sterling from the Flywire community. And intriguingly, the one on the left side, this is the soma, and then it branches in each column of the lobula, in each of the 750 columns of the lobula, and it branches in each column of the medulla. And so how can you do this local computation with such an amacrine cell? So in order to answer this, I did compartmental modeling of CT1 cells. And here you see sort of part of a CT1 cell reconstructed from the EM. And when I injected current into this local process of this compartmental model, then I saw that the excitation remains local. So it seems from this compartmental model simulation that these different little blobs work in electrical isolation from their neighbors. Now, in order to test this prediction experimentally, Matthias Meyer did calcium imaging from the CT1 processes. And again, white noise stimulation, reverse correlation, what he saw that each column here, each blob, has its own little receptive field, as if it were like, a separate columnar neuron, not just one amacrine cell. So that tells us that CT1 is indeed well suited to provide a local inhibitory signal. Now, in order to see whether this actually contributes to directional motion information in T5 cells, Amalia and Matthias killed the CT1 cell by expression of apoptotic genes, namely HIT and REAPER, and then measured the directional tuning of the T5 cells, compared it with control flies. And this is what they got. In black is, again, the control fly, a nice tight directional tuning. And in yellow, you see the directional tuning of T5 cells in the absence of CT1 cell when it was killed. And they also did RDL RNAi experiments where they removed the receptor from the T5 cells, and they saw basically the same, which completely makes sense because CT1 is the only inhibitory neuron. So next question is, so where does CT1 receives its signal from? Well, if we look at the connectome, this is an analysis that Amalia did, we see the different neurons. I already explained to you TM9 here on the preferred side, TM1 and T4 on the central part, and CT1 on the proximal part. Now, within each column, CT1 receives input from TM9, and this is a local connection. So somehow TM9 is acting here, presumably as an enhancing signal for the central one. And at the same time, this signal is then provided to CT1, sort of for sign inversion, to provide null direction suppression. And so, you know, Amalia and Matthias decided to take a closer look at these signals and did calcium imaging and compared it with the signal from other cells. And what they saw was that TM2, as expected for the central signal, is sort of fast, but TM9 is indeed delayed and is following, you know, until a certain point in time here, the time course of CT1. CT1 seems even more slowly than TM9, and as a slow signal is perfectly suited to provide null direction inhibition for these T5 cells. So in summary, while we still don't understand the mechanism of preferred direction enhancement in T5 cells, the null direction suppression is quite unique because it's first of all done by an amacrine cell that has these sort of locally electrically isolated processes. And second, the same signal is used twice, namely the TM9 signal, once, you know, on the preferred side for enhancement and on the null side to, you know, be used as the null direction signal. So that's quite particular. I have no idea how, you know, such a circuit has evolved in evolution. So this brings me to the summary. And I have told you that for most of my career, you know, I've been wondering how the Hasselstein-Reichert motion detector model is implemented in the fly optic lobe. And amongst the many cells, we then found that the T4 and T5 cells are the ones that are first to compute the direction of visual motion. And their functional properties are best shown by these beautiful 4 layers, you know, that correspond to the activity of T4A, B, C, and D cells to rightward, leftward, upward, and downward motion. Then about the question of how they become direction selective, I have shown you that it is a combined mechanism of preferred direction enhancement and null direction suppression, which act in concert to give you a nice tight directional tuning of the T4 and of the T5 cells. Now, with respect to the biophysics of these different mechanisms, I think we're pretty much done. In T4 cells for preferred direction and null direction suppression, we have good evidence for the biophysics and cellular implementation of T5 cells, null direction suppression, but we don't know the mechanism of preferred direction enhancement for T5 cells, but we're working on it. So with this, I want to close and thank, first of all, the Max-Planck Society for supporting, you know, my research and my obsession chasing down the motion detector in the fly visual system for so many years. I want to thank my collaborators at Genelia Farm, Gerald Rubin and Alyosha Nern, for providing these driver lines. Without those driver lines, we couldn't have done any of these experiments. So they were really essential for our research. The same thing is for the connectome work at Genelia and elsewhere. And that really facilitated our research a lot. Simply knowing which cells to test makes life so much easier than if you have to go through, you know, 50 different cell types. So a big thank to those people. That's quite a large team there. But most importantly, I want to thank the motion detectives at our institute. Here you see them all, for their input, for their great experiments that they did, and for you know, sharing their enthusiasm with me to find out how fly neurons compute the direction of motion. Thanks a lot for your attention.
Thank you very much, Axel, for this fascinating and inspiring talk, I dare say. So many questions of mine, but first I would like to let you know that there are already questions appearing in the chat. I made the Zoom room link available in case people want to join us. And as a reminder to the audience, after a first brief round of moderation from my end, that I communicate your questions to Axel, we will move offline and continue in a more informal fashion. So the first question appearing in the chat is from Luisa Ramirez. Thank you very much for the nice talk. If computations in CT1 neurons are local, why to have such a wide field neuron? Would it affect the spatial resolution of T5 when compared to T4?
So that's an obvious question. So why not having 750 columnar neurons? Why have one amacrine cell? Maybe it allows for sort of a common gain mechanism that is, implemented this way. Honestly, I have no idea. It's pure speculation. But there's, to us functionally, there's no difference to having separate columnar neurons.
There are people congratulating you and thanking you for the talk. I just communicate this because you cannot see the chat currently. The second question appearing is from Tom Baden. Tom Baden, many of these biophysical mechanisms presumably depend critically on physical distances and sizes. How then do these insights translate to larger species with bigger eyes and neurons?
That's very good. For me, as a compartmental modeling guy, it was sort of very disappointing to see that you don't need compartmental models, because they are electrically so compact because of the small size of 10 or 15 micrometer dendrites they have. I haven't played around with sort of large-scale models that, maybe blowflies, whether then the space all of a sudden becomes important. They are no longer isopotential then. I also looked at, very hard at any spatial effect, because the processes where the glutamatergic MI9 input synapses onto the dendrite is only like 100 nanometer or 200 nanometers wide. So it's really tiny, 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 yet electrically it doesn't make a difference. So, but you are right. I think, the argument has been made in the vertebrate retina world that, a mechanism that applies to a small species cannot be sort of applied to a large species easily, so to say. I don't know whether there will be sort of important differences if you go to larger species. Maybe not that much, because when the eye grows bigger, the number of facets grows. And so the columns, maybe the column sort of stays at the same size, even in bigger species. So maybe the dendrite of the T4 and T5 cells is not scaling with the body size of the fly, but rather sort of only sort of has a small increment in size. Maybe, I don't know.
Right. Thank you very much for addressing both questions. As we are waiting for some more to appear, people are already joining us here. I will take the advantage to ask some questions that I have. So I was wondering, maybe it's a naive question, but I was wondering whether we know developmentally how all these very specific projections across the dendritic tree occur, whether it's like temporally isolated, so first it's the dendrites and so on, or whether there's selective pruning later on.
George, you couldn't have asked a better question, I have to say, because for decades I said, leave me alone with development. But then a postdoc came to the lab, Jesus, and somehow, step by step, I got into the development, and now we have several people working on development of these neurons. Because how is this spatial specificity achieved during development? One question, or one, so that, this cell takes this from that column, and this cell from that column, and this cell from that column, and the other way, just in a mirror symmetrical way. How is that achieved on Earth? Now, looking at the polarity of the dendrites, we thought that maybe all different subtypes can apply the same rule using their cell intrinsic coordinates, saying that I grab MI4 at the proximal dendrite, MI1 in the center, and MI9 on the tips. And the other one applies the same rule, but has the dendrite just in the opposite way, right? And so the prediction of this model would be that if we somehow can invert the dendrite, then they should wire up just in the new way and then become directional selective in the opposite direction of what they responded to before. And so Nicola Hermann in the lab found that one transcription factor, grain, allows you to invert the direction of the dendrite because it's expressed only in layer 2 and 3 of these neurons and not in layer 1 and 4. And so if you overexpress grain in all the neurons, then you only have type 2 and 3. And they respond to motion just fine according to the new orientation of the dendrite. And so I think that it is the orientation which is the critical part that determines the wiring. And then using cell intrinsic coordinates, the cells could all apply the same rule, you know, MI4 approximately, MI1 here, and MI9 distally. Now, how that is done, I have no idea so far. Whether there's some temporal, processes in place, I don't know. But at least part of the mystery seems to be solved a little bit, at least in my mind.
Right, thank you very much. And a second question before we move offline, if no more questions appear in the chat. So you showed some preliminary data from trying to model these processes and what happens when you play a moving bar of different speeds. I was wondering if you tried also different contrasts or like different luminances to see if the window of null and preferred and how much space they allow for the center would change.
Yeah, I have to say that the stimulus protocol was already pretty big.
Of course, yeah.
It needed the signals of all the 5 different input neurons and the T5 cells to exactly the same stimuli. And so that already takes up about 50 minutes or so. And I could not come up and ask them to also go through different contrasts. So
right.
We simply don't have this data yet, but we might, come up. Now, talking about different contrasts is an important thing because this mechanism has to operate at different contrast levels, of course, and so usually what we see is sort of a contrast saturation, but going to like 5 degrees or 10 degrees, we might get at a critical point, and that would be probably very interesting to have these data, but we don't have them. Sorry.
Thank you. 2 more questions appeared, so let's go through them and then conclude there. So the first one is from Pavan, and I'm sorry if I'm mispronouncing the name. Lovely talk, considering they likely have internal models of the complex natural scene statistics and ground sky motion. How does this tie into our understanding, and where do they exist in the brain?
Yeah. So again, a very good question. And we had one study explicitly addressing this. And that is, if you confront this model the way I have it with natural scenes, it just produces crap. It collapses immediately. Right. And the reason for that is that the cells go from one extreme to the other and all of that. Whereas real T4 cells and T5 cells, as well as their input neurons, don't do that. They adjust the contrast according to the sort of prevailing contrast in the image patch surrounding their receptive field. And so we studied whether it's a process called contrast normalization is also realized in the fly visual system. And we did a very thorough study on that and found that all of or most of these input neurons show this contrast normalization. So they shift their contrast sensitivity to larger contrast according to the prevailing contrast in the environment outside their receptive field. And if we apply in the model contrast normalization to the inputs, everything works nicely again. So yeah, that's an important point. We don't know the cellular nature of that yet.
And last question from Luisa. I think it's inspired from the biophysical findings, but it refers to the model part. The HC circuit has only 2 branches with a unique time delay. What would be the advantage in terms of motion detection to have a 3rd branch?
The third branch is simply to have a tighter tuning. Sort of each process by itself gives you direction selectivity, but together the tuning becomes better, so to say. That's the short answer. In fact, the Riker detector gave sort of a very nice directional tuning through the subtraction process. And for a very long time, we thought, well, you can be quite sloppy there because it's taken care of by the final subtraction on the dendrites of the tangential cells. But T4 cells themselves are already very tightly tuned directionally. And yeah, so that's, I think, the advantage of that.
Right. Then at this point, I would like to thank you once again, Axel, for this fantastic talk. And of course, the Zoom room link is available for people that want to continue offline with us. Thank you very much.
Bye-bye.
So we are officially